Look at your hand. Not a photo of a hand. Yours. Right now. Look at the back of it. The little ridges of skin. The shape of the knuckles. Here's a sentence that sounds insane, and is true. Not one atom in that hand was there 10 years ago. The carbon, the hydrogen, the oxygen, gone, replaced, over and over, your whole life. You are not a thing. You are a process wearing the shape of a thing. So here's the part that should actually bother you. If your body is a machine that rebuilds itself, atom by atom, all day, every day, why does it get worse? A car where every worn part gets swapped for a new one wouldn't age. It would run forever. You are doing exactly that, constantly, and you are still going to get a gray hair, a stiff knee, a wrinkle you didn't have last year. Stay with me for the next half hour. By the end, you'll be able to look at a mirror, a birthday candle, or a bruise healing slowly, and explain in one sentence, with a real paper behind it, why a self-repairing machine still runs down. This isn't a metaphor for aging. It's the actual physics of it, published in a real lab in 2023, with receipts I'll put on screen. Here's why this particular question has bothered thinkers for a very long time, longer than you might expect. It isn't a new worry invented by wellness, marketing, or longevity startups. Aristotle wrote about the natural decline of living things over 2,000 years ago, and every culture that has ever buried its dead has asked some version of the same question you asked the last time you noticed a new line on your face. If the body keeps eating, keeps breathing, keeps healing cuts and regrowing skin, why does the overall pattern still slide, slowly, one direction? For most of human history, the honest answer was a shrug, dressed up as fate, or humors, or the will of the gods. We now actually have a mechanistic answer, sitting at the intersection of a 1,944 physics book, a 1,961 cell biology paper, a 1,963 physics lecture, and a 2023 experiment that finally tested the idea directly instead of just describing it poetically. That's the answer this video is going to hand you. You probably think aging is wear and tear like a shoe sole scraping thin against pavement, or a car's paint fading in the sun, mechanical damage stacking up. Here's the thing, that can't be the real story. Shoes don't eat lunch. Your skin does through you. You have a repair crew running 24 hours a day, enzymes, stem cells, DNA repair machinery, constantly rebuilding what breaks. A shoe has no such crew, and yet you still age. So the wear and tear story is missing the actual mechanism. What fails isn't the material. It's something stranger, and it's written into the second law of thermodynamics itself. Let's give the wear and tear idea its fair hearing first, because your high school biology teacher wasn't wrong exactly. They just stopped one layer too early. Every day, trillions of your cells divide. Skin cells slough off and get replaced roughly every 2 to 4 weeks. Your gut lining rebuilds itself in days. Even in tissues that don't obviously replace themselves like most of your neurons, the proteins inside those cells are being built and broken down and rebuilt on a timescale of hours to weeks. You are less like a stone statue and more like a candle flame. The flame keeps its shape, but the wax burning and the air feeding it are never the same material twice. Go light an actual candle tonight, if you have one lying around, and just watch it for 30 seconds. The flame looks completely stable. Same teardrop shape, same flicker, same warm yellow. But nothing inside that shape is the same from 1 second to the next. Wax molecules are being pulled up the wick, broken apart, combined with oxygen from the room, and thrown out the top as carbon dioxide and water vapor, a constant one-way river of matter, held in a shape by the process itself, not by any fixed material sitting still inside it. That's not a metaphor I'm stretching to fit. It's structurally the same kind of thing your body is. So if the stuff keeps getting replaced, the question isn't why does the stuff wear out, it's why does the pattern degrade if the material doesn't stick around long enough to wear out. For decades, one popular biological answer was what's called the free radical theory of aging, first proposed in 1956 by a radiation chemist named Denham Harman at Berkeley, who noticed that irradiation causes premature aging-like symptoms, that irradiation is known to generate reactive oxygen radicals, and that ordinary metabolism generates those same kinds of radicals even without radiation. His idea: unstable reactive molecules, produced as an ordinary byproduct of breathing and burning fuel for energy chemically damaged cell structures over time. like a slow, internal rusting, and that accumulated damage is what we experience as aging. For decades, this was close to the default explanation taught in biology classrooms, textbooks, and, frankly, in a lot of documentaries that still repeat it uncritically today. It's a genuinely useful idea, and it's not wrong that reactive molecules do cause real chemical damage. But here's the honest part. The part a documentary chasing a clean villain usually skips. When researchers directly tested the theory by breeding animals to produce extra antioxidant enzymes, the exact machinery that neutralizes free radicals, those animals did not, on the whole, live meaningfully longer. If free radical damage were the single dominant cause of aging, cranking up your body's own anti-rust defenses should have been the closest thing to a fountain of youth science has ever engineered. It mostly wasn't. That doesn't mean free radicals are irrelevant. It means aging almost certainly isn't one villain with one weapon. It's several overlapping processes. And the physics question underneath nearly all of them is the same one. Why does a repair system that works most of the time still slowly lose the fight? Here's a small experiment you can actually run tonight, and it's worth doing before you keep watching. Find something metal in your house that's been left outside or in a damp spot, an old nail, a bike chain, a garden tool. Look at the rust on it. That reddish-brown crust is iron atoms that have reacted with oxygen and water to form iron oxide. And it is, genuinely, a form of entropy increase, a more chemically stable, more probable arrangement replacing a less probable one, exactly the same statistical story we're about to walk through with cards and ink. So the wear and tear instinct isn't stupid. It's picking up on something completely real. Rust is real entropy. A scraped knee is real structural damage. Sun-cracked leather is a real chemical breakdown of collagen fibers. Where the instinct runs out of road is your repair crew. A nail has no mechanism to unrust itself, and you do. So the honest question was never, does damage happen? Of course it does, constantly. Exactly as your teacher said, the honest question is, why does a system with an active, dedicated repair crew still lose ground to that damage, when a system with no repair crew at all obviously would? That question takes us out of biology and into physics. Specifically, into a 6-0-year-old physics lecture that most biology classes never mention. In 1963, Richard Feynman gave a lecture about a deceptively simple machine. A wheel with slanted teeth, a ratchet, and a little spring-loaded lever called a pawl that only lets the wheel turn one way. Attach it to a paddle sitting in a box of gas, and the random battering of gas molecules should, in theory, only ever push the wheel forward, one click at a time. One-way motion, pulled out of pure random chaos. Feynman even joked you could tie a string to the axle and lift a tiny weight. His example was a flea for free. Here's the catch, and it's the whole video in miniature. The pawl is also made of atoms. And atoms at any real temperature are jiggling what we call thermal motion, or, if the object is small enough, to visibly dance around in a fluid Brownian motion. So every so often, purely by accident, the pawl itself gets knocked up just high enough for the wheel to slip backward underneath it. Feynman worked out the actual probability of that happening. And if the whole device sits at one uniform temperature, the forward slips and the backward slips balance out exactly. Zero net motion, on average. No free lift for the flea. Why does this matter for your wrinkles? Because your cells run millions of ratchet-like machines, motor proteins, DNA repair enzymes, the molecular machinery that copies your genetic code every time a cell divides. Each one is trying to do the same one-way job the ratchet was trying to do. Take random thermal chaos and turn it into ordered, useful work, a correctly folded protein, a correctly copied strand of DNA. And each one is made of atoms sitting in the same jiggling, bombarding soup of heat as everything else in your body. You cannot build a wall against the chaos out of bricks made of chaos. Let's make this concrete with a deck of cards. Because Feynman's ratchet is elegant but a little abstract, and I promised you something you could explain to someone tonight. A brand new deck comes out of the box in perfect order, ace through king, suit by suit. Shuffle it once, and it's still mostly ordered, with clumps intact. Shuffle it 100 times, and it's a random mess. Here's the honest, slightly uncomfortable physics fact. If you shuffled forever, the deck could, by pure chance, land back in perfect order. Nothing in the laws of physics forbids it. You just wouldn't bet your house on it, because there is exactly one way to be perfectly ordered, ace to king, and there are roughly 8 times 10 to the 67th, that's an 8, followed by 67 zeros, different ways to arrange 52 cards. Order is one outcome among an almost incomprehensible number of possible messes. Disorder wins, not because the universe wants chaos, but because there is so overwhelmingly much more of it available. This, the number of ways a system's insides can be rearranged while still looking the same from outside, is what physicists call entropy. Feynman's own definition, from the same set of lectures. Entropy is the logarithm of the number of ways the inside can be arranged, and still look the same from outside. The second law of thermodynamics says that in any closed process, entropy measured over the whole system never decreases on average. Order becomes disorder. Never, in any experiment ever run, cleanly the other way, for a system left on its own. Look around whatever room you're in right now. If you leave that room alone for a year, no cleaning, windows shut, it drifts toward dust settling evenly, smells mixing, temperature equalizing with the hallway outside. It does not spontaneously tidy itself, sort your socks by color, or refold your laundry. Every physicist who has ever run this experiment on their own apartment can confirm the result. The same logic explains something you've watched 1,000 times without really watching it. Drop a single cube of ice into a glass of warm water, and the cold and the heat mix down to one lukewarm temperature. You have never once, in your entire life, seen a glass of lukewarm water spontaneously separate itself back into an ice cube in warm water. Nothing in Newton's laws technically forbids it. Every individual molecular collision in that glass is perfectly reversible if you ran the film backward, but the number of ways for the energy to end up spread out evenly is so enormously larger than the number of ways for it to end up separated into hot part and cold part that in practice, for any everyday amount of water, it simply never happens. This is the same statistical argument that Ludwig Boltzmann worked out mathematically for gases in the 1870s, roughly 90 years before Feynman's ratchet lecture The Arrow of Time itself. Physicists broadly agree is really just this same statistical argument running at the scale of the entire universe. Now, and this is where a lot of pop science explainers quietly go wrong, you are not a closed system. You eat. You breathe. You radiate heat into the room around you. This matters enormously, and it's the honest answer to the objection you're probably already forming. But I'm alive. I have energy. Doesn't that let me fight the mess? Yes. That is, almost word for word, the question the physicist Erwin Schrödinger asked in 1944 in a short book called What is Life? Not a lab paper, a set of public lectures he gave in Dublin, but one that quietly seeded the entire field of molecular biology. Schrödinger's answer was that a living organism stays organized by feeding on negative entropy, what we'd now more precisely call low entropy energy, like sunlight or the chemical order locked up in food and excreting high entropy waste, like body heat and carbon dioxide. You're not breaking the second law. You're paying it, constantly, by exporting disorder into the room around you so you can keep a little pocket of order inside your own skin. Think about what that actually costs, in plain terms. Every meal you eat is, chemically, a highly ordered structure, a potato, a piece of bread, a steak, all built by a plant or an animal that spent real time and energy assembling those molecules into a specific, low-entropy arrangement. Your body breaks that order apart, extracts the usable energy, and ships the leftover disorder back out as body heat and waste. You are, quite literally, a heat engine that runs on eating order and exhaling chaos every hour of every day, whether you're aware of it or not. A resting adult radiates something in the range of 80 to 100 watts of heat into their surroundings, close to the output of an old incandescent light bulb, just from staying alive and keeping that internal order intact. You are, right now, a slow fire. Give your body credit here, it's an extraordinary machine at this exact job. It has to be, or you wouldn't survive infancy. So the real mystery isn't why does entropy win? Eventually the second law guarantees that for anything short of eating the entire sun. The real mystery is the one Feynman's ratchet already answered for us. Why does the repair machinery itself, the thing doing the paying, slowly get worse at its job? One more thread from that same Feynman lecture, because it answers a question you might already be forming. If every individual collision between molecules is perfectly reversible, run the film backward, and each two-particle collision still obeys the laws of physics exactly, then why can't the whole mixed-up system just unmix itself, given enough time and enough luck? Feynman poses this with two boxes of gas, one full of one kind of molecule, one full of another, separated by a wall. Pull the wall out, and they mix. Run that mixing forward or backward in your head. And here's the strange part. Any single collision, viewed in isolation, looks equally valid running either direction in time. So where does the arrow, the fact that mixing only ever runs one way in the real world, actually come from, if not from the individual collisions themselves? Feynman's answer, and it's a genuinely deep one, it comes from the improbability of the starting condition, not from any law forbidding the reverse. Imagine, he suggests, that the currently observed order in the universe were simply a lucky, temporary fluctuation out of a much larger sea of disorder, the way you could, in principle, deal a shuffled deck and by pure chance get several cards in a row. If that were the whole story, then finding order in one place should tell you nothing about what you'd find if you looked somewhere in the universe you hadn't checked yet. A fluctuation, after all, is a local, one-off accident. But that isn't what astronomers find. Every new star, every new galaxy they point a telescope at, shows the same kinds of physical order, obeying the same laws, consistent with having evolved from a similarly ordered past, not scattered, isolated pockets of order surrounded by unrelated chaos. Feynman's conclusion, which is now close to the mainstream position in cosmology, the order we see isn't a lucky local fluctuation, it's a memory. The early universe itself started in an extraordinarily low entropy, highly ordered state, and everything we call the passage of time, remembering the past but not the future, eggs breaking but never unbreaking, aging happening but never unhappening, is downstream of that one deep fact about how the universe began. You are not just fighting entropy with your dinner and your DNA repair enzymes. You are a small, local eddy in a much larger river that has been flowing the same direction since the beginning of the universe. Bookmark that it's exactly where the next video in this trilogy picks up. There's one more piece of that same 1963 lecture I want to give you, because it's the single most direct answer to anyone who says, sure, but couldn't you just build a perfect sorting machine, one that never lets the mess back in? Feynman raises exactly this, using an old thought experiment from the physicist James Clerk Maxwell. Imagine two boxes of gas at the same temperature, connected by a small hole with a tiny door, and sitting at that door is a hypothetical demon, really just a very small machine watching the molecules go by. Every time a fast, hot molecule approaches from the left, the demon opens the door and lets it through to the right. Every time a slow, cold molecule approaches, it keeps the door shut. Do this long enough, and the right-hand box gets hotter, the left-hand box gets colder, with no outside energy put in, which looks, on the face of it, like a flat violation of the second law. Here's Feynman's resolution, and it's the same idea we've been circling the entire video, just from a different direction. The demon itself is a physical object, made of finite, physical parts, gears, a spring-loaded door, whatever you build it from, sitting in the very same warm gas it's watching. Because it's finite, it can't dump the heat it accumulates from constantly watching and reacting to molecules fast enough. It heats up. And once it heats up, its own moving parts start to jiggle from thermal motion, the same Brownian jiggling that made the ratchet's-pawl slip, and the demon starts making mistakes, letting a slow molecule through by accident, or missing a fast one. Eventually, it's shaking so much from its own accumulated heat that it can no longer reliably tell a fast molecule from a slow one at all. The demon doesn't fail because the idea was flawed. It fails because any machine capable of sorting order out of chaos must itself be built from atoms, and those atoms are subject to exactly the disorder the machine is trying to sort against. If that sentence sounds familiar, it should. It's the ratchet-and-pawl again, wearing a different costume. And it's your DNA repair enzymes, your protein-folding machinery, your immune system's molecular sorting mechanisms wearing that same costume a third time. Every demon your body builds to keep itself organized is, itself, made of the very thermal chaos it's fighting. One quick, honest detour, because the word information is about to carry a lot of weight in this video, and I don't want it to feel like a poetic flourish borrowed from a computer science class. In 1961, the very same year, Hayflick published his cell division limit. An IBM physicist named Rolf Landauer showed something remarkable. Erasing a single bit of information in any physical system, anywhere in the universe, always costs a minimum, calculable amount of energy, released as heat into the surroundings. Not usually, not as an engineering inconvenience, as a hard floor set by the laws of thermodynamics themselves. His famous one-line summary of this was information is physical, meaning a bit stored in your laptop, in a strand of DNA, or in the exact pattern of chemical tags sitting on top of your chromosomes, isn't an abstract idea floating free of physics. It's a physical arrangement of atoms, and rearranging or erasing that arrangement has a real thermodynamic cost and a real thermodynamic vulnerability to noise. This is exactly why epigenetic information in the Sinclair paper isn't a soft metaphor stapled onto real biology for a good headline. The pattern of chemical tags controlling which genes are switched on in a given cell is, physically, information sitting on a molecular substrate, and like any physical information, sitting in a warm, jiggling cell for decades, it is exposed to exactly the same thermal noise Feynman described in his ratchet lecture and Landauer quantified in his own. The reason a 2023 cell biology paper and a 1963 physics lecture and a 1961 information theory paper are all, underneath the jargon, describing the same fight is that they are the same fight, physical order, sitting in a warm universe, slowly losing precision to thermal noise, whether that order is a strand of DNA, a magnetic bit on a hard drive, or a pole holding a ratchet in place. Before we get to the 2023 study, I want to show you something that makes all of this less abstract, and it isn't a mouse. It's a real, rare human condition called Hutchinson-Gilford Progeria syndrome. Children born with this condition carry a spontaneous mutation in a single gene called LMNA, which builds a protein called lamin A, one of the structural scaffolding proteins that holds the inside of your cell's nucleus organized, the way tent poles hold up canvas. The mutated version, sometimes called progerin, folds wrong and gets stuck in the nuclear membrane, and it physically distorts the shape of the nucleus and disorganizes how DNA is packaged inside it. The result is not a slow biological process playing out over decades. It's the same category of process structural information inside the nucleus, losing its organization compressed brutally into childhood. Affected children typically show hair loss, joint stiffness, and hardening of the arteries by early childhood, and the average life expectancy is around 14 to 15 years, with most deaths caused by heart attack or stroke from severe, early atherosclerosis, the same cardiovascular disease that, in the rest of us, shows up decades later. This isn't offered as an emotional shortcut. It's evidence. Progeria is, in effect, a natural experiment the rest of us never had to volunteer for. Break one structural gene that helps keep nuclear architecture organized, and you get an accelerated preview of hallmarks that otherwise take a normal human lifetime to accumulate. It's a hard, honest reminder that what we're discussing tonight isn't a comforting metaphor about card games. Somewhere, right now, there are children living through a compressed version of exactly the physics we're about to walk through in full. This is where I want to be very careful, because this is exactly the kind of claim that gets people burned by content that sounds profound and turns out to be unsourced. So let's do this properly, with real names, a real year, and a real journal on screen. In January 2023, a team led by Jaehyun Yang, with David Sinclair as senior author, published a study in the journal Cell that did something genuinely new. They didn't just observe that aging correlates with a loss of biological information, they built a system to cause it, on purpose, and watch what happened. Here's the setup, in plain language. Every cell in your body carries the same DNA, the hardware, the actual sequence of genetic letters. But which genes get switched on or off in a given cell is controlled by a separate layer sitting on top of the DNA called the epigenome. Think of it as the software that tells a skin cell to behave like skin and a neuron to behave like a neuron. Sinclair's team engineered mice with a system they called ICE-inducible changes to the epigenome that let them deliberately trigger small, repairable DNA breaks over and over without changing a single genetic letter. The DNA sequence, the hardware stayed completely intact, and yet the mice aged faster, measurably. Their tissues showed the classic hallmarks of old age: skin thinning, hair graying, muscle weakening, cognitive decline on standard mouse tests, all while their actual genetic code, letter for letter, was untouched. Why would that happen? Because every time a cell repairs a DNA break-even, a repair that fixes the sequence perfectly, the repair proteins have to physically relocate to the break site. That's a real, physical process, and it's not perfectly precise. Some of those chromatin-modifying proteins don't make it all the way back to their original position afterward. Multiply that tiny positioning error by trillions of repairs over decades, and the software which gene is switched on, in which cell, at what level, gradually blurs. Not because the hardware failed, because the read and repair process itself, like Feynman's pawl, is carried out by machinery that is never perfectly precise, because it's made of the same jiggling atoms as everything else. Here's the part that made this study a genuine landmark, rather than just a confirmation of an old idea. Sinclair's team could partially reverse it. Using a partial set of the so-called Yamanaka reprogramming factors, often abbreviated OSK, they restored some youthful gene expression patterns and some youthful function in affected tissues. If aging were simple, permanent damage to the hardware, you couldn't reverse it by resetting software. The fact that partial reversal worked is strong evidence for the information loss model, specifically not just where, but a blurring of instructions that can, at least partly, be resharpened. I want to be honest about what this does and doesn't prove, because that's the difference between evidence and a good story. This is one mouse study, using one engineered method of accelerating epigenetic drift. It's a landmark piece of evidence for aging involves loss of epigenetic information, not a complete, closed case that this is the only cause of aging. Most gerontologists would tell you aging is almost certainly caused by several overlapping mechanisms at once, not one single villain, which brings us to a second real study. Because good evidence stacks, it doesn't stand alone. Back in 1961, Leonard Hayflick and Paul Moorhead discovered something that, at the time, overturned the accepted wisdom that cells grown outside the body could divide forever. Instead, they found that normal human cells in a lab dish divide a finite number of times, commonly cited as roughly 40 to 60 divisions, before they stop dividing altogether and enter a state called senescence. This is the Hayflick limit, and it's tied to structures called telomeres, protective caps on the ends of your chromosomes that shorten a little with every division, like the plastic tip of a shoelace slowly fraying. Eventually, the cap gets too short to protect the chromosome, and the cell locks itself out of dividing a built-in safety brake, not a random breakdown. Here's the part that turns this from a curiosity into another piece of completing rather than contradicting the picture we're building. Your cells actually do carry an enzyme called telomerase, which can rebuild those shortened caps. So why doesn't your body just run it constantly in every cell and stop the shortening altogether? Because a cell that can divide without limit, ignoring its own safety brake, is one short step away from the textbook definition of a cancer cell. Most of your ordinary cells keep telomerase switched off, specifically because unlimited division is dangerous, not because the enzyme doesn't exist. The Hayflick limit isn't a design flaw the universe forgot to patch. It looks, more and more, like a deliberate trade-off your own biology makes, except a slow, statistical decline in exchange for a much lower chance, most of your life, of a single rogue cell dividing forever and killing you outright. Even the flaw in the system is doing a job. And a third piece, more recent and, to me, one of the most beautiful comparative studies in this whole area. In 2022, a team at the Wellcome Sanger Institute, led by Alex Kagan, sequenced DNA from intestinal tissue 208 individual intestinal crypts from 56 individuals across 16 different mammal species, everything from mice to giraffes. They were hunting for somatic mutations, small copying errors that accumulate in ordinary body cells, not the reproductive cells passed to offspring across species with wildly different lifespans. What they found, the rate at which these copying errors pile up per year varies enormously between species, and it scales inversely with lifespan. Short-lived species like mice rack up mutations fast. Long-lived species like giraffes and humans accumulate them far more slowly per year. But, and this is the striking part, when you multiply each species' yearly mutation rate by its typical lifespan, The total number of mutations accumulated by the end of a natural life comes out roughly similar across species. Nature seems to have tuned the typo rate of different animals' copying machinery to roughly match how long that animal is built to live. 3 separate, independent lines of evidence: an epigenetic drift experiment in mice, a 40-plus-year-old cell division limit backed by telomere biology, and a cross-species mutation accumulation study, all pointing at the same underlying picture. Aging tracks a slow accumulation of copying and repair imperfections in a body that is nonetheless working constantly and mostly successfully against exactly that outcome. Here's what I'm not going to tell you, that this closes the book. Gerontology today talks about aging in terms of a whole set of interacting hallmarks: genomic instability, telomere shortening, epigenetic alterations, loss of proteostasis, mitochondrial dysfunction, cellular senescence, and several others, and the honest, current scientific position is that these hallmarks feed into each other rather than one single cause explaining everything. The Sinclair information theory framework is one of the most actively discussed models in the field right now, and it's genuinely well evidenced for the mechanisms it directly tests. It's also a young idea by the standards of a field that's been arguing about this since Hayflick's cells stopped dividing in a lab dish in 1961. Any documentary that tells you the single, final answer to why we age has already stopped doing science and started doing marketing. What all of these competing hallmarks share, though every single one of them, is this same underlying physics problem: a repair or maintenance system built out of atoms, trying to hold a low-entropy pattern together against the same thermal jiggling that Feynman put a name and a number on in 1963. So here's the reframe, and I want you to actually sit with it for a second. You didn't fail to stay young. You are winning every single day against the second law of thermodynamics, a law that governs steam engines and shuffling cards and the death of stars, a law that has never once been violated in the history of physics, and you're winning it using machinery built from the same jiggling, imperfect atoms that the law is trying to scramble. A wrinkle isn't proof your body broke. It's a scoreboard. It's the tiny, accumulated residue of tens of trillions of successful repairs. For every failed one, there were an almost unimaginable number that worked silently your whole life without you ever noticing. Compare that, honestly, to the deck of cards from earlier. A freshly shuffled deck reaches full disorder in about 100 shuffles. Left alone, with nothing feeding it order, it stays disordered forever. Your body is not doing that. Your body has been fighting off complete disorder, successfully, for decades, using nothing but food, sunlight, sleep, and molecular machinery built from ordinary atoms, atoms that have no special immunity to the very jiggling that's working against them. Every year you're alive, past childhood, is a year that fight was on net, still going your way. The wrinkle showing up now doesn't erase that record. It's the current scoreline in a game you've already won, by a wide margin, for a very long time. What do you actually do with this? Not a supplement pitch, the honest, current state of the science. If aging really does involve information loss on top of pure structural wear, then the research this points towards still early, still actively debated among scientists, and not something I'm going to oversell to you, is less about stopping decay and more about maintaining fidelity in the repair process itself. Things like DNA repair capacity, protein quality control, and, in Sinclair's specific framework, epigenetic stability. None of that is a pill you can buy this year. What is genuinely, boringly true today, backed by decades of ordinary evidence, is that the things that reduce the rate of new damage, regular movement, adequate sleep, not smoking, protecting skin from UV, which is itself a major driver of DNA damage in skin cells, reduce the raw workload on the very repair systems this whole video has been about. You're not fighting entropy by wishing. You're fighting it, quite literally, by giving your repair crew fewer typos to fix in the first place. There's an entire modern research field built around treating aging itself, rather than any one disease of aging. As the thing worth studying directly, it's often called geroscience, and its working assumption is close to what we've spent this whole video building, that many of the diseases we treat separately in old age, heart disease, many cancers, dementia, joint disease share a small set of underlying biological processes, and that intervening on those shared processes, rather than chasing each disease one at a time after it appears, might be a more efficient place to aim research. That framework doesn't hand you a product to buy. What it does hand you, right now, honestly, is a much better way to evaluate anything that claims to be an anti-aging breakthrough. Ask which specific underlying process it claims to target. Ask whether that claim has been tested in a controlled, published, peer-reviewed way. And ask in what organism, at what dose, over what time. If a claim can't answer those questions, it isn't evidence. It's marketing, wearing a lab coat. I also want to head off the version of this idea that tends to run wild online. Aging is information loss does not mean we found the aging switch or reversal is available now. The mouse study we walked through used a specific engineered method to accelerate epigenetic drift and a specific partial reprogramming method to partially reverse it in a controlled lab setting, in mice, over a defined and relatively short window of their lives. Translating that safely into an intervention for an entire adult human body where you cannot risk resetting cell identity so far that a liver cell forgets it's a liver cell is a completely different, much harder engineering problem, and one that serious labs are still actively working through rather than one anyone has solved. If you ever see a product marketed as reverses aging by restoring your epigenetic information without a named, dated, peer-reviewed human trial attached to it, that's a claim borrowing the vocabulary of real science to sell you something the science hasn't actually delivered yet. That's not cynicism. That's the same evidence standard we just used to praise the Sinclair paper applied consistently, including to claims you might actually want to be true. One more honest, useful thread before we close this out. Your repair systems also have their own built-in maintenance routines, and stressing them lightly, on purpose, seems to matter. Physical exercise, for instance, triggers a cellular cleanup process called autophagy, literally self-eating, where cells break down and recycle their own damaged components, including misfolded proteins and worn-out cellular machinery. Periods of fasting appear to trigger a similar cleanup response. None of this stops the second law. Nothing can. But it's a genuinely physical, mechanistic reason why the boring advice, move your body, don't constantly overload it with excess fuel it doesn't need, sleep enough for your cells to run their nightly maintenance shift, keeps showing up in the actual literature instead of just in wellness marketing. The advice is boring because the physics underneath it is real, not because nobody has found the exciting version yet. Here's what I keep coming back to. The universe, left to itself, wants to be a boring, uniform soup. Every particle spread evenly, every temperature equalized, nothing happening. Physicists have a name for that end state, equilibrium. And equilibrium, for a living thing, is just another word for death. You are the opposite of that. You are 7 billion atoms that have somehow arranged themselves so precisely that they can wonder about their own arrangement. For 70, 80, maybe 100 years, you are a small, brilliantly organized exception to the way the rest of the universe behaves, and you're maintaining that exception using repair machinery, that is, itself, made of the very chaos it's fighting. That's not a small thing. That's the most improbable event most of us will ever be part of, and it's happening inside your own skin right now. We are not competing with Feynman tonight, or with Schrödinger, or with the Sinclair-lab. We're completing them, taking a 60-year-old lecture about a wheel and a pawl, a nearly 100-year-old idea about negative entropy, and a paper published in 2023 and putting them next to each other so you can see they were always describing the same fight. Your body isn't failing a test it was supposed to pass perfectly. It's running the only kind of process the universe allows for something built out of jiggling atoms trying to stay organized. An imperfect, ongoing, mostly winning argument with the second law of thermodynamics. Every gray hair is a receipt from a battle you're still, on balance, winning. Look at your hand again, the same one from the start of this video. Every physical atom in it will be gone and replaced again within a few years, same as it has been your entire life. What won't be gone, what has actually persisted since you were an infant, is the pattern, the incredibly improbable, actively defended arrangement that makes that pattern recognizably, continuously you. Physics doesn't have a mechanism for stopping the second law. No paper, no supplement, no breakthrough headline will ever hand you that. Because it isn't available to hand out, it would mean rewriting the most tested law in all of science. What physics does have, and what this video handed you, is the actual mechanism for why the fight is so hard. why your body is nonetheless so good at it for so long, and why the newest evidence points toward maintaining precision rather than repairing damage after the fact as the most promising place to keep looking. So here's a question with no wrong answer. When you noticed your first gray hair, or the first morning your knees actually announced themselves, did it feel like losing something, or did some small part of you register it as evidence you were still in the fight? Tell me, in a few words, in the comments, what's the first sign of aging you remember actually noticing? If you want the receipts every time real papers, real years, real journals, no unsourced studies show that's exactly what the physics storyteller is built to do. Subscribing to the physics storyteller means you stop guessing about how the universe works and start seeing it. This is actually the first of 3 videos on entropy and time on this channel next. We go from your body to the universe itself and ask a stranger question. Does yesterday still exist somewhere? That's coming up on the physics storyteller. Nature only uses her longest threads to weave her patterns, so that each small piece of the fabric still reveals the shape of the whole tapestry. Your aging hand is one thread. Tell someone that tonight. Watch their face.